Today we know that Epstein-Barr virus causes cervical lymphadenopathy, sore throat, fever, malaise, that we call infectious mononucleosis. But in 1940s, they did not used to know what is causing this enlargement of lymph nodes, sore throat, fever, which virus is causing this acute illness. So what they did at that time in 1940s, they took serum from human being and they mixed with the sheep blood. They isolated serum from human being and they mixed with the sheep red blood cells, and those sheep red blood cells agglutinated. So they thought there is something in the human serum that is causing agglutination of the sheep red blood cells. Generally it should not cross react because we are Homo sapiens and sheep is different species. So generally our antibody should not agglutinate other species red blood cells like horse red blood cells, sheep red blood cells. It should not happen normally. But there is something in the serum that is causing agglutination of the other species red blood cells. So they call it heterophile antibody. Hetero means different species. Phile means loving. Phile means loving. So that antibody was loving different species. That's why they call it heterophile antibody. And they commercialized that test, heterophile antibody test, for rapid detection of Epstein-Barr virus. But this is not always positive in Epstein-Barr virus. If you get this heterophile antibody test, it does not mean that you have only Epstein-Barr virus. You can have CMV, you can have HIV, you can have autoimmune disease, because in those conditions also some heterophile antibodies are secreted. So they later developed Epstein-Barr virus specific antibodies. But before discovery of Epstein-Barr virus specific antibody, heterophile antibody was widely used.